The last couple of lectures are about nucleotide metabolism, 44 and 45. Learning outcomes for this one, we will describe the purine nucleotide synthesis, pyrimidine synthesis, and then the regulation of the nucleotide synthesis in the first part of the lecture. So there are two main ways of the nucleotide synthesis. They can be synthesized de novo, or commonly we call it de novo synthesis, that is from the beginning or from the start. So we start with some raw material like amino acids, sugars, carbon dioxide, and ammonia, and we make a new nucleotide. And the second is known as the salvage pathways. These nucleotides can be salvaged from the old DNA, RNA, and the cofactor degradation as well. So the second one is more or less like a recycling pathways. So first up, the de novo synthesis, or synthesis from the beginning, using the raw materials, and it's approximately the same in all organisms and all kinds of species which has been studied. This de novo synthesis is almost the same. And phosphoribosyl pyrophosphate, or PRPP, is made from the ribose 5-phosphate, which is the activated ribose. And very important points to remember is that glutamine provides most of the amino groups in this de novo synthesis. Glycine is the precursor for the purines, which are the big nucleotides, the one with two rings. Aspartate is the precursor for purines and pyrimidines, both of them. The nucleotide pools except the ATP are usually kept very low in the cells, so that the cells have to continuously synthesize them whenever there is a need for synthesis. But ATP, as we all know, is a very common molecule inside the cell, so it is always present in abundance. But apart from that, rest of the nucleotides, the concentration is kept very low. So we saw the synthesis of ribose 5-phosphate when we were discussing the carbohydrate metabolism during the pathway, which is the PP, or pentose phosphate pathway. We start off with glucose. There was an oxidative phase in which we synthesized this sugar, ribose 5-phosphate, at the end of this one, and this sugar is going to be used for the synthesis of nucleotides, which is eventually incorporated into the DNAs and the RNAs. So the source of ribose 5-phosphate is PPP. So the first step in the synthesis of these nucleotides is going to be the synthesis of PRPP molecule, or 5-phosphoribosyl 1-pyrophosphate, shortened as PRPP, which we synthesize from the ribose 5-phosphate sugar, which we just saw we get from the pentose phosphate pathway. So in the first step, this sugar, ribose 5-phosphate, will be converted into PRPP, and the PRPP synthetase is the name of the enzyme. A highly regulated allosteric enzyme it is, so it has to be controlled and regulated. Now here we have a structure of a typical purine, in which we can see different atoms. As I told you in the previous lecture as well, a purine is made up of two hydrocarbon rings fused together. One is a 6-carbon or a 6-cornered ring, and the second one is a 5-cornered ring. And here we can see where the particular atoms come from in this purine molecule. The glutamine will provide 2 nitrogens, one for each ring. Formate provides a couple of carbons. 1 carbon dioxide is also used in the synthesis, so that carbon will be incorporated as well. 1 carbon comes from aspartate, and another one comes from glycine, which also gives us a couple of carbons as well. So this is the source of the atoms that we find in the purine molecule. So here we have the general overview or the summary of the purine biosynthesis basically, and it begins with the PRPP molecule that we saw in the previous slide that we generated from ribose 5-phosphate sugar. So the actual synthesis begins with this PRPP joining with the glutamine amino acid, and this is the committed step of the cycle, the control point of the cycle, you can say the most important step, and in which PRPP and glutamine join together to give us the phosphoribosylamine, and the purine ring will build up following addition of 2 carbons and 1 nitrogen from the glycine. So in the next step, glycine will be added up in this molecule, and the purine ring will be built. And very important point to remember is that the first intermediate with the full purine ring, in which the ring is completely formed, is known as the inosinate, or we can also call this molecule inosine monophosphate, shortened as IMP. So IMP is, you can say, a precursor, is also known as a parent purine nucleotide, from which we can synthesize both adenine and guanine, which are the actual purine nucleotides that we are synthesizing. So here we can see that the first reaction in the diagrammatic view, PRPP, joins up with the glutamine amino acid, and the product is 5-phosphoribosylamine, and this is the committed step, the rate limiting step of the whole pathway, the control point. So if we have to control the whole pathway, if we want to activate it or slow it down or inhibit it, we will have to control this enzyme at this first reaction step. And later on, we add up a glycine, and the ring keeps on building up. You don't really have to go into the details of all of these reactions, but there are plenty of reactions in the pathway, and we are slowly synthesizing this ring of the purines. So as I said, it's a long pathway. The FGAR, which has got one ring present in it, will be converted into the FGAM and eventually to AIR, but still not the full ring has been made yet. So the pathway continues. This aminoimidazole ribonucleotide, or shortened form AIR, uses some energy from the ATP, is converted into CAIR, and the CAIR is going to be converted into succinyl aminoimidazole carboxamide ribonucleotide, or SAICAR. So at the end of these 11 reactions is where we get the inosinate or IMP molecule, and as I said, this one is a very important intermediate during the pathway, as the full purine ring has been formed in this molecule or in this intermediate. So eventually from this IMP we can generate the guanosine or adenosine, whichever we want. It's a monophosphate molecule. One phosphate is attached along with a 5-carbon sugar as well, which is the typical for any nucleotide. So here we have the final couple of steps to generate the AMP or the GMP molecule from the parent purine nucleotide, which is inosinate or IMP. If we have to synthesize the AMP, in the first step we will have to add up aspartate to it. Energy will be provided by the GTP molecule. We convert it into an intermediate adenylosuccinate, and eventually the lyase enzyme will break this adenylosuccinate, take the fumarate away, and we get our final product, which is AMP. If we have to synthesize the GMP molecule, inosinate will first be converted into xanthylate, or shortened form XMP, by addition of water to this molecule, and eventually the glutamine is going to be added up, and we get the GMP molecule, or guanylate. Next up, we have the regulation of this purine biosynthesis pathway. And the main thing to remember is the whole regulation largely consists of the feedback inhibition. And you guys should be familiar with the feedback inhibition by now. It's the kind of inhibition in which the product loops back to one of the reactions and inhibit one of the important enzymes. There are four major mechanisms by which we regulate this purine biosynthesis. By regulating the glutamine PRPP amidotransferase enzyme, which is inhibited by the end products IMP, AMP, and GMP. Excess GMP can also inhibit the xanthylate enzyme, and the PRPP synthesis is also inhibited by the ADP and GDP. So any of these final products or one of the intermediates as well can loop back to one of the main enzyme and cause the inhibition of that enzyme. And the most important or the committed step is this amidotransferase. So that is usually the control point of the pathway. Next up, we have the de novo synthesis for the pyrimidines. Pyrimidines we discussed in the previous lecture are the small nucleotides like cytosine, thymine, and uracil. So for synthesizing these pyrimidines, we need three main raw materials: aspartate amino acid, PRPP, which we just saw is you can say the activated ribose, or is uh, the product of the ribose 5-phosphate sugar. And the third thing we need is carbamoyl phosphate, which is another molecule. So carbamoyl phosphate, which is required. So we saw aspartate is a common amino acid usually found inside the cell. PRPP we can synthesize from the ribose 5-phosphate, whereas this carbamoyl phosphate will be synthesized by the enzyme carbamoyl phosphate synthetase 2, or CPS2 is the short form of the enzyme in the cytoplasm. Now this enzyme is pretty similar to an enzyme CPS1, which we saw in the urea cycle, which catalyzed the first step of the urea cycle. Now there is an important point of difference between the purine and pyrimidine synthesis. So unlike the purine synthesis, pyrimidine synthesis proceeds by first making the pyrimidine ring and then attaching it to the ribose sugar. Whereas we just discussed the purine synthesis. In the purine synthesis, we first attach the sugar, and the purine ring was made at the end of 11 reactions. But for pyrimidine synthesis, we make the ring first and then the reactions continue, and eventually we add up the sugar to it. So we start off the synthesis of pyrimidines by this first committed step, or the most important reaction for this pathway, in which we join aspartate and carbamoyl phosphate and get a carbamoyl aspartate molecule. The name of the enzyme catalyzing the reaction is aspartate transcarbamylase, shortened as ATCase, and this is the rate limiting step for the enzyme, and the reaction is the committed step. Once this reaction is done, it's almost certain that the whole pathway will proceed. This carbamoyl aspartate is converted into dihydroorotate and eventually to orotate by the hydrogenase enzyme. So the synthesis continues. Orotate is converted into orotidylate, and then we get the uretylate, the UMP, which can be considered as the parent pyrimidine nucleotide, and this is one of the pyrimidines that we are going to synthesize. So first up, always UMP is synthesized. So UMP is the first possible pyrimidine, as it is written here in the slide as well. So the pathway will continue if we do not want the UMP and we want some other nucleotide. So UMP can be phosphorylated to UTP molecule, adding up 2 phosphates to it. 2 ATPs will be utilized in this reaction. So UMP is converted into UTP, and eventually the cytidylate synthetase enzyme will convert it into CTP molecule, which is the cytidine triphosphate. which is the second one of the pyrimidine nucleotide. Next up, we have the regulation of the pyrimidine synthesis, and just like the purine synthesis, the main mechanism of pyrimidine biosynthesis regulation is also by the feedback mechanism. We just saw that the CTP is the final product of the pathway, and when we have high concentration of this final product, it will loop back to the very first reaction of the pathway, which was catalyzed by aspartate transcarbamylase. It will inhibit this enzyme, so the whole pathway will be stopped or inhibited, whereas ATP accelerates or activates this pathway. So an important point to note, guys, is that the nucleoside monophosphates must have to be converted into nucleoside triphosphates in order to be incorporated into the nucleic acids. So in most of the cells of the body, the phosphorylation of AMP to ADP, that is to the diphosphate form, is catalyzed by the enzyme adenylate kinase. And the ADP is phosphorylated to ATP, usually by the glycolytic enzymes or through the oxidative phosphorylation. And this ATP will always serve as a phosphate donor for any other nucleotide. So any nucleoside monophosphates can be converted into diphosphates with the help of kinases, and as I said, ATP will be the phosphate donor, and similarly any nucleotide diphosphate can be converted into triphosphate with the help of kinases as well, and usually ATP is the donor for that reaction as well. Another important slide here is about the ribonucleotides and the deoxyribonucleotides. So always remember the general rule that ribonucleotides are the precursors for the deoxyribonucleotides. So from the ribonucleotides, we will always make the deoxyribonucleotide. So the example of the reaction shown is an NDP, which is a diphosphate nucleotide, and it will be converted into deoxyribonucleotide. And the main differences we discussed in one of the previous lectures as well. The difference between the ribonucleotide or deoxyribonucleotide is simply an oxygen atom that is present right here in the hydroxyl group. We take this oxygen out, and the ribonucleotide becomes deoxyribonucleotide. So ribonucleotides are incorporated in the RNAs, as we know, and the deoxy ones are incorporated into the DNAs. So that is why we need the deoxy ribonucleotides to be made from these. And an important thing to remember, which I almost forgot, is the name of the enzyme which catalyzes this reaction, ribonucleotide reductase. So the type of reaction is the reduction reaction. Two hydrogen atoms will be donated by the NADPH during the reaction. So as we saw in the previous slide, reduction of the ribonucleotides into deoxy form is done by the ribonucleotide reductase enzyme, and the NADPH serves as an electron donor. So the NADPH will convert the glutathione into the reduced form, and that reduced form is used for the glutaridoxine, and that glutaridoxine eventually converts it into the ribonucleotide reductase, the active form. So this is like a cascade of reactions that are involved. The main molecules to remember are NADPH, which is serving as the electron donor, and this will activate the enzyme ribonucleotide reductase, and this enzyme catalyzes this reduction reaction of converting the NDP to DNDP, and the substrates are always ribonucleoside diphosphates or the DP varieties for this reaction. Next up is something that we call a roundabout pathway, just because of the shape of the pathway. And in this pathway, we synthesize the DTMP molecule, or thiamine deoxythiamine monophosphate. and the immediate precursor for this one is DUMP. So we can start off the process with CDP or UDP. First convert it into the deoxy form by the reaction that we just saw in the previous slide by the reductase enzyme, and then a kinase will use to convert these into the triphosphate form. So DP is converted into DCTP, and the UDP deoxy form is converted into the DUTP, the triphosphate form. Then we have to take the phosphates out of it. So UTP is used to take two phosphates out. So the deoxy uridine triphosphate is converted into the monophosphate form, and eventually the final reaction of the pathway, which is catalyzed by thymidylate synthase, and an important enzyme to remember, converts the DUMP into the DTMP form, or thiamine monophosphate. So this is the pathway of synthesizing the DTMP molecule. So last up is the lecture number 45, which is the nucleotide metabolism part 2. And in this part, it's basically the reversal of the first part. So these will be the breakdown processes: the purine breakdown, pyrimidine breakdown, and the purine salvage pathways, which are the recycling pathways for the purines. So this is an important slide. There's some general summary, or you can say the overview of the catabolism of the purines. So all the purine degradation always leads to one product, which is uric acid, and uric acid can be the final product, or it can further go on, but you can say the most common intermediately final product is uric acid. Sometimes we can convert the uric acid into some other products as well, but mostly the catabolism of purine stops at this molecule, which is uric acid. Now another important thing to remember is all the ingested nucleic acids are degraded into nucleotides. So any kind of food that we eat, if it had any nucleic acids in it, any DNAs and RNAs, they will always be broken down to nucleotides in our body by the pancreatic nucleases. These are the enzymes that break them. And then we have intestinal phosphodiesterases as well in the intestine. So these are the two enzymes that help in the digestion of the nucleotides, and most of these ingested nucleic acids will be broken down and eventually excreted from our body. We do not use the foreign nucleic acids for any kind of function inside our body. So we break it down and degrade it and excrete it out of the body. Now group-specific nucleotidases and some non-specific phosphatases are also present, which degrade the nucleotides into nucleosides. So we saw in the previous lectures, nucleotide is made up of three parts basically. One is the nitrogenous base, one sugar, and one phosphate. So that is what we call the nucleotide. If we take the phosphate out of it, it is known as a nucleoside. So nucleotidases will be the enzyme that takes the phosphate out. Then further degradation or further breakdown will take place. The nucleoside will be broken down into its two parts, that is the nitrogenous base and the sugar part. And this is done by nucleosidase. And it can also be broken with the help of phosphate, or phosphorylase will be the enzyme in the second case, but the products are the same, base and the sugar as well. Sugar this time will be in the form of ribose 1-phosphate. So this is the short pathway for the catabolism of purines. Not many reactions in there. Whether we and the most important thing to remember, whether we start with AMP or GMP purine, the final product is going to be the uric acid. And as we saw in the previous slide, there are different enzymes to break these nucleotides down. If we start with the AMP, we have the nucleotidase enzyme, which takes the phosphate out and convert AMP into adenosine, which is the nucleosidase. So phosphate has been removed. Next step is remove the ammonia from it by the deaminase enzyme, and we get the product inosine. And the third and last one, in which we take the sugar out as well, and only the nitrogenous base is left, which is known as hypoxanthine. This one is also done by nucleosidase. Eventually, this hypoxanthine is converted to xanthine, which is the keto form, and eventually the second step done by the same enzyme, xanthine oxidase, it is converted into the uric acid, which is our final product. So xanthine oxidase is the most important enzyme for this metabolic pathway, which converts the xanthine into uric acid. Now, on the other hand, if we start with the GMP, a similar process is going to take place. First, we take the phosphate out of the nucleotide, it becomes nucleoside, the name is guanosine. Then we take the sugar out, it becomes guanine, which is the nitrogenous base, and guanine is going to be converted into xanthine with the help of deaminase enzyme, which takes the ammonia or NH3 out of it. And from the xanthine, it's the same reaction as it was in the case of AMP molecule, xanthine into uric acid with the help of xanthine oxidase enzyme. And this is the most important enzyme of this pathway, basically, that you have to memorize. Now, as I said in the beginning, this uric acid can stop at this point as well, or in certain instances it can be converted into other molecules as well. So degree of further oxidation of the uric acid is basically organism dependent. It depends on the organism in which it is being produced. So the primates, birds and reptiles usually excrete this nitrogen waste in the form of uric acid. Most mammals can convert it into another molecule, allantoin, and they excrete it. Some bony fishes convert it into allantoate and then excrete the nitrogenous waste. And in some amphibians and cartilaginous fishes, it is converted into urea. And eventually we have marine invertebrates in which urea will be converted into ammonium ion as well. So these are different molecules that we can produce from the uric acid. Do remember, uric acid is a waste product which has to be excreted out of the body. So depending upon the organism, if it can excrete it out or not, if it cannot excrete uric acid, it will convert it into some other molecule that it can easily excrete. It keeps on, or the pathway keeps on continuing until the organism can excrete that molecule out of it. Next up, we have a disease that is known as gout, and gout is a result of too much uric acid inside the body, excess uric acid. And in this disease, we see painful joints, especially of the joints of the foot, that is, of the toes. And the reason for the painful joints is the deposits of sodium urate crystals. So the high uric acid levels crystallizes and form sodium urate crystals. These crystals deposit in the joints and cause pain in the joints and also inflammation. Primarily this disease affects the males, so males are more common. And the reason of increased uric acid is usually the under excretion or over consumption of the fructose. One of the two reasons can be there, that the body is not excreting enough uric acid out of the body, or the body is making more uric acid than normal inside the body. And the treatment involves avoidance of the purine rich foods like seafood and the livers, and also avoidance of fructose as well. So all of these substances we have to stop eating from and have to avoid these foods. And the most commonly used drug for treating this gout is allopurinol. and this drug inhibits the main enzyme of the purine degradation pathway, which is xanthine oxidase. So that was the main enzyme of the catabolism of purines, and this allopurinol inhibits this enzyme, so the breakdown is basically stopped. Next up we have the catabolism of pyrimidines, and the most important thing to note is whichever pyrimidine we start off, the most common ones are cytosine and thiamine. So if we start from cytosine, we will first convert it into uracil, and we take this pathway, and at the end of many reactions we get the final product acetyl CoA. And if we start with thiamine, the final product is going to be succinyl CoA. Now the point to remember is that both of these, acetyl CoA and succinyl CoA, are intermediates of the citric acid cycle. So these intermediates can enter the citric acid cycle, and we can get energy from them. The catabolism of pyrimidines produces the intermediates of the citric acid cycle, which are acetyl CoA in case of cytosine and uracil, and succinyl CoA in case of thiamine. Now the last part is about the salvage pathways. So salvage pathways, as I said in the beginning as well, are the recycling pathways. So any of the purines and pyrimidines which are old inside our body, we can recycle them through these salvage pathways, and we can make new purines and pyrimidines from them. And for these salvage or recycling pathways, we get any free bases which are released in the metabolism. They are going to be reused. So we break down certain things, we get these free bases, and we can use them to synthesize the nucleotides. Now two important enzymes are there in the salvage pathway: adenine phosphoribosyl transferase, or APRT is the short form, and the second one is HGPRT, or the full form hypoxanthine guanine phosphoribosyl transferase. So these are the two main enzymes, APRT and HGPRT. The short forms are there on the next slide in the diagram. Now very important thing to remember is this HGPRT enzyme, if it is missing or it is not performing its function properly due to the lack or deficiency of this enzyme, a disease occurs which is known as Leishman syndrome. And in this Leishman syndrome, which mainly affects the brain, there are some neurological impairments seen, and finger and toe biting behavior is also seen in such patients. So here is the diagram for the salvage pathways. As I said, two key enzymes are there, APRT and HGPRT. Deficiency of HGPRT results in the Leishman syndrome. Now in rapidly dividing cells like the bone marrow cells, and in young children in which the growth is present, another enzyme, adenosine deaminase, is also important, which is shortened as ADA.